Hello, welcome to PCAP's Native Prairie Speaker Series. My name is Caitlin Mroz, and I am the Stewardship Coordinator with Saskatchewan Prairie Conservation Action Plan, or PCAP. Today, Kirsten Palmier, Masters of Science candidate at the University of Regina, will be talking about pathogens on the prairies, assessing health of native Saskatchewan bumblebees. Every month, PCAP asks someone to present either in the form of a webinar or an in-person talk in the Saskatchewan community on anything to do with native prairie conservation or species at risk. Join us on November 15th at noon for a talk about bat landscape use during migration by Erin Swedberger, Master of Science candidate at the University of Regina. On November 29th at noon, Brandon Berta, Master's of Science student at the University of Regina, will be talking about sharp-tailed grouse leks in southwest Saskatchewan. To register or find out more information about these webinars, please visit the PCAP website, www.pcap-sk.org. I'd like to take a moment to note that financial support for today's webinar is provided by TransCanada Corporation, Canada North Environmental Services, Environment and Climate Change Canada, Eco-Friendly Sask, Information Services Corporation, Ranchers Stewardship Alliance Inc., SaskTel, and Wildlife Habitat Canada. In-kind support for today's webinar has been given by the University of Regina. A reminder to all of our listeners out there, if you have any questions during the presentation, please feel free to type it into the question section of the webinar dashboard at any time during the presentation. Questions will be answered at the end of the webinar. Now, a bit about our presenter. Growing up in Saskatchewan, Kirsten has always had an appreciation for the outdoors, which has led her to complete her Bachelor's of Science from the University of Regina in 2017. Her passion for nature lies with the protection and conservation of native Saskatchewan species. Throughout her undergraduate degree, Kirsten has been fortunate to volunteer for organizations that focus on the conservation of native prairie species, such as the Nature Conservancy of Canada, the Regina MAPS Monitoring Aviation Productivity and Survivorship Program, and Parks Canada. These volunteer opportunities range from mist-netting songbirds in Regina to tracking black-footed ferrets in Grasslands National Park. She's also had the pleasure of working at the Royal Saskatchewan Museum as a summer student since 2015 under the supervision of Dr. Corey Sheffield, which consequently has led her to pursue her Master's of Science research at the University of Regina. Kirsten's current area of research, co-supervised by Dr. Sheffield with the RSM and Dr. Andrew Cameron at the University of Regina, focuses on health and conservation of native bumblebees, specifically the presence of pathogens within the province of Saskatchewan. And with that, I'd like to turn it over to Kirsten.
Thank you, Caitlin, and thank you for inviting me to talk today, and thank you everybody for tuning into my webinar. Today I would like to talk to you about my current master's research. Oh, sorry, I just see. There we go. Today I would like to talk to you about my current master's research, Pathogens on the Prairies, Assessing Health of Native Saskatchewan Bumblebees. Bees are arguably one of the most important species on the planet. They are said to be ecosystem engineers, meaning they have the ability to drastically transform landscapes. When bees are present in an ecosystem, they are said to increase species richness, which is the number of different species that represent an ecological community, landscape, or region. In addition, bees can also increase the landscape heterogeneity, which is considered a key determinant for biodiversity. How do they do this? Bees are able to accomplish this by means of pollination. In agricultural landscapes, when bees are present, they are said to increase outputs, lower production costs, and increase the quality of the products being farmed. Before we get going here, I'd like to take the opportunity to discuss native bee diversity. There are an estimated 20,000 different species of bees that occur worldwide. There are roughly 4,000 different species of bees in North America alone. Actually, all of the bees found on this slide are native to Saskatchewan. Bees can come in a variety of sizes, from small bees, like this Hylaeus shown here, which is approximately 4.5 millimeters, to larger bees, like bumblebees shown here, which are approximately 15 millimeters. They can also exhibit varying colors, like this blue orchard bee shown here, or colorful maculations shown here by this nomada. Different species of bees have different lifestyles as well. Some bees live a social lifestyle or otherwise have a colony, like bumblebees. Some bees live solitary lifestyles with varying degrees of parental care, like this Hylaeus and this longhorn bee or Melizodes. Others are kleptoparasites, like this nomada, stealing pollen from other bees. Some bees are ground nesting and can either live in their own cavities or in old burrows of small animals. Other bees can be found nesting in pre-existing cavities and pithy stems. Some bees are generalists, meaning they pollinate a variety of plants, while others are specialists, pollinating only a few variety of plants. Please note that I am not talking about imported pollinators, although they are agriculturally important. My talk will focus on native bees, specifically bumblebees. But what exactly is a bumblebee? Bumblebees are part of the Apidae family. There are 45 species of bumblebees that occur in Canada, with 27 species found in Saskatchewan, including 5 species of the parasitic subgenus Cytherus. This lovely specimen is a bumblebee called Bombus borealis. It's a common bee found in Saskatchewan. Bumblebees are generalist foragers, feeding on the nectar and pollen of many different types of flowering plants. Bumblebees are proficient pollinators of many agricultural crops as well as native flora. Their fuzzy bodies, ability to forage during undesirable conditions such as cooler climates and lower light densities, and their ability to buzz pollinate make them effective pollinators. Bumblebees are eusocial, meaning colony members cooperate in brood care and have a division of labor among overlapping generations. A single female, queen or foundress, will start a colony each spring. Once a colony is established, duties such as brood care, gathering of nectar and pollen, and cleaning the nest will be divided and performed by the daughters, leaving the foundress committed almost exclusively to egg laying. Bumblebee colonies are smaller than honeybee colonies, with only up to a few hundred individuals. Sorry, this next slide is taking a second to load. Although all bees are vegetarian, feeding off the pollen and nectar of flowering plants, different species of female bees have different means of carrying pollen back to their homes. Some solitary or parasitic bees lack these specialized structures entirely, or will carry pollen and nectar in an internal structure called a crop. Leafcutter bees, or megachili, which are seen here, possess a scopa on their abdomen, which is a large, dense mass of branched hairs that allows them to carry and gather pollen. On the longhorned bee, or this Melizodes that's shown down here, the scopa is found on their back legs. In bumblebees, shown here, the scopa is replaced by a specialized structure called a corbicula or pollen basket. Actually, if you look really closely, you can see the remnants of collected pollen that are still stuck to its leg. A corbicula is a polished cavity found on the hind leg that is surrounded by dense fringed hairs. After the pollen has been collected, it is pressed onto the pollen basket by female bees with comb and brush-like hairs found on the inside of the legs. The evolution of pollen basket in bees has been linked to the development of more complex social behaviors that are seen within the Apidae family. There has been a lot of recent media attention to declines in bee populations globally, but what is the current status of bumblebee populations? A study by Kola and Packer compared museum-caught specimens captured in Ontario during the early 70s to specimens captured during the early 2000s. The researchers collected bees at the same sites and followed the same sampling protocols. On the x-axis, you will find the different species of bees, and on the y-axis, you will find the relative abundance of each. In the top corner, the black bars represent the samples caught during the 70s, and the sample size for the study is listed as well. Here, the red bars represent the specimens captured during the summer months of 2004 to 2006. Again, the sample size is listed in the top right-hand corner. First, I would like to point out that the change in relative abundance of a few species, Bombus impatiens, found here, and Bombus terricola. The relative abundance of Bombus impatiens has tripled since the 70s, where Bombus terricola does not appear to occur at this site any longer. And lastly, I would like to point out the variation in sample sizes. During the 1970s, there were nearly 3,400 specimens captured, while during the sampling season in the 2000s, roughly only a third of the original sample size were captured. So not only did the relative abundance of bees change, but the population sizes have dropped as well. The following species of bumblebees have been experiencing dramatic declines throughout North America: Bombus terricola, Bombus occidentalis, Bombus affinis, Bombus pennsylvanicus, and Bombus bohemicus. Bombus bohemicus is a part of that parasitic subgenus, the Cythrus. My talk today will focus on two species of interest, Bombus terricola and Bombus occidentalis, which are actually both shown here foraging on flowers. So this one would be terricola, and this is occidentalis. Bombus terricola, otherwise known as the yellow-banded bumblebee, occurs throughout North America, though it is most commonly found in the east. Despite their large range throughout Canada and the northern US, its abundance has been declining in parts of its range. For this reason, they have been listed by the Committee on the Status of Endangered Wildlife in Canada as special concern. Bombus occidentalis is the sister species to Bombus terricola. They are so closely related that they were once thought to be the same species. Opposite to its sister species, Bombus occidentalis have an entirely western distribution. Since the 1990s, populations in Western Canada have undergone dramatic declines, going from one of the most abundant bumblebees to one of the rarest. For this reason, this species has been listed as threatened by COSEWIC. Commercialization of bumblebees began in Belgium in 1987 when colonies of Bombus terrestris were used to aid in the pollination of tomato crops. An added benefit of managed bees is that you can manipulate their life cycles to use them for pollination services year-round, where in the wild, bumblebees will follow an annual cycle. It is estimated that over 1 million colonies are exported globally per year. Bombus impatiens, known as the common eastern bumblebee, is native to southern Ontario and Quebec in Canada. Bombus impatiens is a popular choice for North American growers as it can easily be managed and distributed for commercial greenhouse crop pollination. Bombus impatiens has proven to be a very successful candidate for commercial trade as they are very adaptable. Unfortunately, due to their adaptability, they have become a widespread species with an expanding range, largely due to accidental escapes and subsequent establishment after being introduced. For this reason, they have been listed by the International Union for the Conservation of Nature as least concern. Currently, scientists believe that there is not one main factor that is causing declines in bees, rather a combination of stressors acting upon the bees at once. Some of the potential causes or stressors for declines include habitat loss, competition for resources, pesticides, climate change, and pathogens, although my research will primarily focus on pathogens. What is the difference between a pathogen and a parasite? Parasites and pathogens can both cause diseases in their hosts, although in order to be classified as a pathogen, it must cause a disease within its host. In addition, parasites depend on the host organism for their own benefits. Typically, parasites are larger or macroscopic, and some examples of bee parasites include conopid flies, which lay their eggs within the abdomen of a bee, which is actually shown up here, or mites that can be found outside of the bee or internally. Pathogens are typically smaller organisms that live internally and will always cause a disease to their host. Some examples of bee pathogens are deformed wing virus, with an individual showing signs of an infection here, and nosema, which I will discuss in more detail. Nosema species are obligatory intracellular parasitic microsporidian related to fungi and predominantly found within bumblebees. There are 3 known species that affect bees. Nosema bombi predominantly affects bumblebees. Nosema apis affects honeybees, and Nosema ceranae can affect both. Nosema spreads orally between individuals through spores found in pollen or from bee excrement. It infects the host Malpighian tubes, which is a type of excretory and osmoregulatory system similar to our kidneys, which can also be found in the gut and the lumen. High levels of Nosema infections have been found within declining bee populations and are thought to be a main contributor to global bee declines. The effects of Nosema include decreased survival rates in males, reduction of sperm, changes in colony behavior such as dysentery, and increased mortality rates. Trypanosomatids are a diverse group of flagellated protozoans with agricultural and medical importance. They can cause diseases in humans, such as sleeping sickness that is caused by the vector of a tsetse fly, as well as diseases in plants and bees. Crithidia bombi was the first described species of trypanosomatids to infect the guts of bumblebees. In 2010, Tegnaso and Schmidt-Hempel discovered a close relative of Crithidia bombi, Crithidia expoeci. Historically, all diagnosed cases of Crithidia lumped together both Crithidia bombi and Crithidia expoeci, and therefore there is little known about the distribution of this newly defined pathogen. Unlike many other trypanosomatids, Crithidia follow a simple life cycle, as they do not require a vector for transmission between hosts. Crithidia are transmitted within colonies on contaminated surfaces and food, as well as between colonies through mechanisms such as flower sharing. Bumblebee queens infected with Crithidia are castrated, which lowers the overall genetic variability of populations in addition to reducing the fitness of founding queens in the spring. Worker bees infected with Crithidia exhibit reduced foraging efficiency, impairing their ability to learn the color of awarding flowers. This change in foraging behavior negatively affects colony success as well as plant reproductive success. Our last pathogen of interest is the Neogregarine Apicystis bombi. Apicystus bombi is a parasitic alveolate that infects bees. It is believed to have a cosmopolitan distribution, meaning it is likely to occur globally. Historically, Apicystus was diagnosed with low infection rates found in bumblebees and sporadic infection rates found in honeybees. Apicystus bombi was first discovered in overwintering queens in Ontario, with Bombus vagans, shown here, being the most heavily infected species. It has now been recorded in 20 different bumblebee species worldwide. The effects of an infection include a disruption and reduction of fat tissue, which is necessary for founding queens to start a new colony, increased mortality rates in workers, and communication conflicts between queens and workers. In order to demonstrate the complicated interactions between managed bees and wild bees and their pathogens, I'm going to use a figure from Greystock and others. First, let us begin with parasite facilitation. When bees are exposed to higher levels of stress, they are more susceptible to infections, independent of any transmission between the two species. In other words, if the addition of managed species increases competition for food, wild bees may become more susceptible to naturally occurring pathogens. Spillover happens, seen up here, when a reservoir population, in this case managed bees, transmit disease to native populations. Pathogen spillover can introduce new pathogens into an ecosystem or introduce a more virulent strain of pathogens to populations that cannot tolerate them. Pathogen virulence may differ between species, but closely related species have a greater potential to transmit between them. Spillback, or otherwise known as reverse spillover, occurs when naturally occurring pathogens found in wild bees transmit their pathogens to managed populations. The naturally occurring pathogens may become more virulent within the managed colonies as the managed bees can tolerate the pathogens better, largely due to the favorable conditions in which they are reared, resulting in asymptomatic infected bees. A study by Greystock and others in 2013 tested for the presence of pathogens in UK managed bumblebee colonies and the pollen that they're supplied with. It must be noted that these colonies were claiming to be parasite and pathogen-free and were ready for distribution. On the x-axis, you will find the managed bumblebee species and the pollen that they've been fed. On the y-axis, you will find the proportion of colonies that were infected. There are a few important things I want to point out. The first is that both commercially reared subspecies are carrying a multitude of different pathogens. And secondly, the pollen that they're being fed is carrying not only bumblebee parasites, but also honeybee pathogens as well. A study by Kola and others in 2006 tested the hypothesis of whether or not there is a higher proportion of individuals testing positive for select pathogens at greenhouse sites. On the x-axis, you will find the sample sites, with the greenhouse sites being on the left and the non-greenhouse sites being on the right. Again, on the y-axis, you will find the proportion of individuals that tested positive for the select pathogens. As you can see, there were not only a higher proportion of individuals testing positive for pathogens within the greenhouse sites, but the diversity of pathogens increased at the greenhouse sites as well. Which brings me to my project. I have two research objectives, the first being a hypothesis that we will find a higher proportion of individuals testing positive for pathogens within closer proximity to greenhouses. Saskatchewan offers us a unique research opportunity because the ranges of Bombus occidentalis, shown in the blue, and Bombus terricola, shown in the purple, overlap within the province. There have been documented declines of both of these species in North America. However, the bumblebee populations appear to be stable within the province. Redcliffe is a small community near to Medicine Hat that is largely known as the greenhouse capital of Alberta. Because of this, we will be using Redcliffe as our predicted epicenter for pathogens. This summer, I sampled bees within closer proximity to Redcliffe and moved outwards to test whether or not there was a higher proportion of individuals testing positive for pathogens within closer proximity to our ground zero site. I captured 50 bees at each sample site using aerial nets, and each individual bee was stored in their own Eppendorf tube. At our ground zero site, I captured bees within Redcliffe, outside of greenhouses, and 2 sites that were at least 10 kilometers away. Currently, there is no known knowledge of what bumblebee pathogens are present in Saskatchewan. To determine this, I traveled around the province and visited 8 out of the 11 ecoregions in Saskatchewan. The ecoregions I visited were the cypress upland, which is found in this little guy here, the mixed grassland, the moist mixed grassland, shown here, aspen parkland, shown here, the boreal transition, shown here, the mid-boreal lowland, shown here, and the Church River upland, shown in the light green. In each ecoregion, I had 6 sample sites. 3 sites were found within provincial parks to reflect natural pathogen loads. The other 3 sample sites were in urban and rural areas, so either larger communities like Regina or smaller communities like Green Lake. So needless to say, we covered a lot of ground this summer. This brought my total sample sites up to 53, with a total sample size of 2,650 individuals. To preserve our specimens and their DNA, we kept these bees in 100% ethanol and kept them frozen until further analyses. Once the specimens were ready to be processed, I dissected them to screen for any parasites, such as mites, fly larvae, etc., and removed their fat bodies, Malpighian tubes, and guts to screen for pathogens. Because the pathogens I'm testing for are transmitted orally, I'm avoiding cross-contamination between individuals by screening each bee's internal organs. After their guts have been removed, I'm extracting their DNA using a modified salting out protocol by Sambrook and Russell. After the cells have been lysed or broken open, there are extracellular components such as proteins and DNA-degrading enzymes within the solution. We can remove these contaminants at high salt concentrations as they dehydrate, lose their solubility, and precipitate to the top of our mixture. The precipitated contaminants can be removed by centrifuging the samples, and we are left with our pure DNA. So this is actually pure DNA in a buffer solution shown here. It's kind of cool to see. To test for these pathogens, I'm using a molecular tool called a polymerase chain reaction, or PCR. PCR was developed by Carrie Mullis in 1983. It is a relatively inexpensive and simple method for amplifying particular segments of DNA that can later be visualized on an agarose gel. For this example, I'll be explaining the process of PCR using DNA that has been extracted from a bumblebee, which is this little green double helix here. The little blue squares represent nucleotides, so extra A, C, T's, and G's that we will need to produce more copies of our amplified gene. Other important players in the PCR process are primers, or small fragments of genetic code that will bind to our DNA fragments to amplify our target gene, which is shown in red or pink, and a polymerase, which is not shown, an enzyme that is used to assemble our desired DNA molecules. The first step in PCR is denaturation of our DNA, so breaking apart that double helix. This step requires high enough heat to split the DNA into two strands without degrading the DNA. In the second step, or annealing, the primers bind to their targeted fragments of DNA. In the third step, or elongation, seen here, is where the nucleotides are arranged complementary to our broken strands of DNA. These three steps will be repeated to produce many copies of our targeted sequences, in this case, the pathogen sequences, which can later be visualized on an egg roast gel. In the summer of 2016, I did a preliminary screen for pathogens within the province. 2 sample sites with a total of 30 individuals were captured and revealed that our bees are in fact carrying pathogens. During my survey, 3 species of bees tested positive for select pathogens. This species is Bombus trinarius, a common bee found within the province, and it had 2 individuals test positive for Apocystis bombi, 1 individual test positive for Crithidia bombi, and 1 individual test positive for Crithidia expoeci. Out of the Bombus terricola that I caught, shown in the middle, 1 individual tested positive for Apocystis bombi, 2 individuals tested positive for Crithidia bombi, and 5 individuals tested positive for Nosema bombi. 2 of these individuals exhibited co-infections of 2 of these pathogens. And lastly, out of the 3rd species I captured, Bombus vagans, seen here, 2 individuals tested positive for Apocystis, 4 tested positive for Crithidia bombi, 2 tested positive for Crithidia expoeci, and 1 tested positive for Nosema bombi. 1 individual displayed a co-infection, testing positive for Apocystis, Crithidia bombi, and Nosema bombi. These results may be the first record of this newly defined pathogen, Crithidia expoeci, in Canada. Some other cool things I found while I was dissecting were the following. In my Turnarius species, I found 3 canopid fly larvae. In my Bombus terricola species, I found 1 canopid fly larvae as well as 4 males that had an enlarged proventriculus, which is a structure that filters pollen and nectar within the bee's digestive tract. We are unsure what caused this swelling, but it appears to be that some sort of yeast that has proliferated within the structure. In my Bombus vagans species, I found an additional 3 canopid fly larvae. My second research objective is to update our current knowledge on bumblebee populations. We will pay special attention to species of interest, Bombus terricola and Bombus occidentalis, as well as document if Bombus impatiens' range is expanding. Historically, Bombus impatiens is not found outside of its eastern range. But after the introduction as a managed pollinator in British Columbia, there is documented evidence of its establishment. We know that Bombus impatient colonies are being used in Redcliffe, but we should not expect to find them outside of the greenhouses. In addition to screening for pathogens across the province, I also left out blue vein traps, which are these lovely guys right here, a passive and effective trapping method filled with propylene glycol to preserve specimens at all of my sample sites. This data will help to interpret the status of our bumblebee populations, including the occurrence of certain species as well as their relative abundance. 24 specimens of Bombus impatiens have been captured outside of greenhouses during a period from 2015 to 2018. Although there is no clear evidence if they are establishing, as only workers and males have been captured, there are still potential threats that this may cause to surrounding native bee populations. Those potential threats include competition for resources, integration with related species, changes in seed sets of native plants, pollination of exotic weeds, and the transmission of pathogens. So what I will be busy working on for the next year or two will be the following. For my pathogen research, I will continue to dissect and process my specimens. I will be looking for the presence or absence of select pathogens, Nosema, Crithidia, and Apocystis, using microscopy and conventional PCR. We are looking for any geographic patterns of pathogen prevalence, and if there is a link to higher pathogen loads within closer proximity to greenhouses. In addition, because we are testing every bee individually, we are looking for patterns of pathogen prevalence within certain closely related species, such as Bombus occidentalis and Bombus terricola, as they are experiencing dramatic declines elsewhere. For my occurrence and abundance data, I will continue to ID specimens captured in blue vein traps and netting. We are also going to assess the current status of bumblebees found within the province and compare to our older museum specimens. Again, we are looking for any geographical patterns between the ecoregions. This data can also be used for future studies on bumblebees, as each individual will be contributing valuable knowledge after being added to the Royal Saskatchewan Museum's collection. I'd like to thank my supervisors, Dr. Andrew Cameron, and the Cameron Lab members at the University of Regina, as well as Dr. Corey Sheffield and members of the Sheffield Lab for their continued support. I would also like to thank the lovely folks at the Logan Bee Lab in Utah for their expertise and help in detecting bee pathogens. I'd like to thank the Ministry of Parks, Culture, Recreation, and Sport for allowing me to collect in various parks throughout the province. And lastly, I'd like to thank the Saskatchewan Ministry of Agriculture and the Canada-Saskatchewan Growing Forward 2 bilateral agreement. And now I'm ready for questions.
Thank you very much, Kirsten. That presentation was really interesting. I've always heard about bees on the media and I haven't really understood what's been going on. So thank you for updating us about bees in Canada. To all of our listeners out there, if you have any questions, please feel free to type it into the question section of the webinar dashboard. And it looks like we've got a few questions coming in. The first one is, have you ever been stung during your research?
Oh, yes. Although bumblebees are fairly docile, they don't like being shoved into a 1.5 milliliter tube. So yeah, definitely have been stung when I haven't been paying attention, and it does hurt.
One of our listeners would like to know, what's the effect of commercial crop herbicides on bee population? That's a listener named Luca.
Okay. Unfortunately, I haven't looked much into pesticides because I've been focusing more on pathogens. But I thought I remembered reading something about their ability to withstand pathogen loads with different pesticide use. But I don't know enough of that to comment on it, unfortunately.
Okay, thank you. We have a few listeners who are interested to know if you've done any work with bumblebeewatch.org for data distribution, if you're able to use the sightings that are recorded there. Yeah, have you heard of that organization before?
I have. I've actually contributed a couple of sightings myself. It's a nifty little app that you can use. And sometimes when I'm out in the field, it helps with field identifications too. As whether or not we're going to be using that data for our study, I'm still unsure. We've talked about it, but we haven't set anything in stone yet, but it would be an interesting way to include more citizen science into science as well. I think that app's great.
Awesome. That's actually the next question is, would you be accepting specimens collected by citizen scientists, assuming that they've been collected into alcohol and frozen as you have done?
Right. I mean, any data, I'm sure the museum would definitely like to have as many samples as they can for the collection, which I can definitely use for my research. So I would say, sure, why not? But as far as my my current project and research, that might have been my only sampling season, and so I'm not sure if I would be still collecting data next year. But the museum would definitely take specimens.
Awesome. Thank you for that answer. We've got lots of questions coming in, so this is great. One of our listeners was wondering if you could show the slide again of the reasons for bee declines. That's a listener named Melody.
The potential causes?
Yes.
Yeah, of course. I'll just zoom to that here.
Perfect.
So, yeah, I just want to mention too that I mentioned it in my talk, but I really want to stress that there isn't one cause that's causing all of these declines, or that's what researchers are believing, is that it's more of a combination of all of these stressors acting upon them at once. So because they don't have the same habitat, or because they're competing for food, because of pesticides, because of climate change, they might not be able to withstand their natural pathogen loads. So just a combination of everything is probably the reason for the declines, not just one factor.
Perfect. Thank you for that answer. And Melody typed in again, thank you. One of our listeners, Tracy, would like to know, she's wondering if we should also be concerned about the spread of pathogens from honeybees to native bees, given the increasing popularity of beekeeping.
Yeah, so there is the potential for the bees to spread pathogens between each other. My recommendation would be having honeybees in an environment, if you're not using them for a certain reason and you think that you're putting them out to save bee populations, you might be doing the opposite. So I would avoid putting colonies out unless you're planning on harvesting for honey, or using them as pollinators in your field. I don't know if that answers the question.
I think so. Thanks. A listener named Ryan has a really interesting question here. If someone wants to do a survey of bumblebees on their property using a live capture with SweepNet, and if there's a chance of finding bee terricola or bee occidentalis, does the person require a permit, given that these are listed species?
I don't think that they do. I guess you do. I needed a permit to collect bees in provincial parks. If you are planning on collecting them, if it's just for observation, I wouldn't kill them. You can release them afterwards. They won't sting you. They'll just kind of fly away. If you're collecting them for your own collection, I would avoid collecting too many of them if possible, especially in the spring when the queens are out, and then later in the season when the queens are establishing their new nests for or finding new hibernation spots. But I don't believe that you need a permit to collect bees on your own.
Thank you for that answer. Regarding the slide that you're showing right now, the potential causes of decline, a listener named Stephen would like to know what the percentage for each percentage cause for decline is, or are they synergistic effects?
Yeah, I'm not entirely sure what the different percentages are. I don't know if that's something that scientists can necessarily determine at this point. Because there are so many different variables, it's unclear.
Yeah, there's a lot going on there.
Yeah.
Another listener would like to know, is all of your sampling destructive? She understands that the parasite research would require destructive sampling, but for the identification part, do you have to use destructive sampling methods, or is there another way?
Definitely. Because I'm contributing all of the specimens that I've captured into the museum collection, it is a destructive means of sampling, but you can capture them when you're IDing them. I recommend taking photos. Another non-destructive way of sampling for the bees as well is you can take off one of their legs and you can test them. You sequence it so you can tell what species you have. But unfortunately for my research, it is destructive sampling. Fortunately, there are a lot more insects than there are other things. So it's kind of a drop in the bucket for how many species or bees are still out there.
Okay, thank you.
Mm-hmm.
A listener named Stephen would like to know, has anyone started to study the effects of glycophosphates on bees? Or glyphosate, sorry. Misread that.
Again, that is not my area of expertise, so unfortunately I haven't really looked into that. But I imagine that that anything that has to do with pesticides or agricultural research, that they're probably looking into those things because they are important pollinators.
Thank you. And a listener named Vanessa would like to know if you have any identification keys that you would recommend for native bees.
Well, we've been using a book by Paul Williams. It's Bees of North America. And I find it really helpful. It's a little bit confusing because there are some structures that you have to see under a microscope in order to determine exactly what species you're dealing with. But I think that that's definitely a great start, as well as any of those apps. So the Bumblebee Watch app, I find sometimes very helpful in the field, as well as an iNaturalist app where you can take a picture, and this goes for plants, insects, and other mammals. You can take a picture of what you're looking at, and an algorithm will kind of show you the closest looking species, and it's actually fairly accurate. At least it would be able to get down to family level.
Perfect. And then to all of our listeners out there too, if you are interested in seeing this presentation again and learning more about the identification of bees, it will be uploaded to YouTube as well, to the PCAP YouTube channel. And that's youtube.com/user/skpcap. I know for me, who doesn't know anything about bees, I'll definitely be going through it again and learning some of the types, because I've seen them before and I've always wanted to know. So I guess this might be our last question here. A listener named Luca has a loaded question, and he's wondering if the world could survive without bees.
I would say no. Well, they pollinate so many different types of plants that a lot of the food that we eat is dependent on bees for pollination. And although there are some foods that are wind pollinated, they still tend to do better with bees around. So I think bees are really important and that we need to keep them safe.
Thank you. Thank you very much for that. And that looks like, and he also says thank you too. This looks like all the questions that we have today. So thank you very much for the really interesting presentation. I know I got a lot out of it. And thank you to all of our listeners today for taking the time to tune in and watch our Native Prairie Speaker Series. So with that, I'd like to wish everyone a great day and happy Tuesday.
Thank you, everyone.
Thank you. Bye.